In this video, we're going to continue to talk about how RNA can be changed. And so in our last video, we talked a lot about splicing of mRNA, but mRNA can go through other changes as well, such as changing of bases, deletion, and insertions of nucleotides. One common example of this is the methylation of A residues, and the most extreme example of this is in certain protozoa, several hundred U residues will be added after transcription is already complete. And how do we modify the bases in RNA? Well, we have special machinery known as guide RNAs or gRNAs. And what they'll do is that these guide RNAs will be complementary to the part of the mRNA that needs to be modified. So they'll bind there, and then they're going to direct the alteration with the protein that does the modification. So just to show you an example of this, we have some substitutional editing where we can take one base and just change it into another. And this is thought to increase protein diversity. Example would be an enzyme deaminates C to make a U, or A to make inosine, which is read as G. And a real example of this is with ApoB, chylomicrons, which we already talked about this semester. And two different ApoB proteins are made from the single gene. Those are APO48 and APO100. And the way that we can get two proteins from one gene is that we do that enzyme catalyzed deamination. And in APO48, we change a C to a U. Normally, for APO100, that residue CAA, which will make glutamine. But if we change that C to a U, we make a stop codon. That stop codon is making this very shorter version of our APO enzyme. And the enzyme that carries this out is cytidine deaminase. So after we have created our mRNA, we've capped it, put a tail on it, modified any bases, we finally have to get it out of the nucleus before we can make a protein out of it. Remember, our ribosomes aren't in the nucleus, they're in the cytoplasm. And this has to be highly selective because the nucleus has a lot of RNA, and only 5% of the RNA that we make will ever leave the nucleus. So how can we make sure to only export the RNA we want to? Well, let's remember that mRNA will be associated with a lot of proteins, such as the capping protein, the poly A tail protein, a protein we're going to look at called the exon junction complex, EJC, and other proteins called SRs and HNRNPs. So what is this EJC? Well, during the splicing process, EJC is added about 20 to 24 nucleotides upstream of the splice junction. And this will sit there, and along with all the other proteins that are with mRNA, it is thought that these proteins serve as an ID to the cell of this mRNA needs to be exported out of the nucleus. And so what will happen is that this mRNA and all the proteins that are associated with it will go to the nuclear pore complexes, or NPCs. Now, the NPCs are relatively large pores, and they allow free diffusion of any molecule up to 40 kilodaltons, but the mRNA and the proteins associated with are too big to leave by themselves. And so what happens is that the proteins that are associated to mRNAs have export signals, as in they need to leave the cell. So they will go over and they're recognized by a specific protein called DBP5, which is a helicase, RNA helicase. So we're going to, DPB5 will be associated with the NPC. Our proteins will go and interact with DPP5, and this helicase will kind of just pull the mRNA out of the nucleus into the cytosol. And while doing this, while pulling it out, the proteins associated with the mRNA, a lot of these proteins will be stripped off, and then they can go and interact with a different mRNA molecule. Any other proteins that make it through this process will be removed when the mRNA goes to interact with the ribosome. And after a while, our mRNA will eventually be destroyed in the cytoplasm. And this is a highly regulated process, as you can imagine, because we want to control how much protein we make. So the degradation varies with the identity of mRNA, what type of cell you're in, and your developmental stage. Now we're going to move on to a different type of RNA. Let's talk about rRNA, or ribosomal RNA. E. coli has 3 different types of rRNA called 5S, 16S, and 23S. These are specified by 7 different operons, and these are nearly identical, but the introns are slightly different in sequence. Now, before these are mature rRNA, the primary transcript do need to be processed. And this actually occurs, the first step occurs while you're still making the rRNA. And you're going to have cleavage by enzymes called RNase 3, RNase P, RNase E, RNase F. And you can see here on this image, each arrow is pointing to where a different cleavage site is happening with the primary transcript. You can see originally this is made as one long RNA sequence, but after cutting it up, you get pre-16S, pre-23S, and pre-5S. So in addition to our introns being cut out, we also need to remove the 5' and 3' nucleotides. This is carried out by RNases D, M16, M23, and N5. But this cutting of the 5' and 3' only happens after our pre-RNA is already associated with the ribosomal proteins. Remember, a ribosome is made out of RNA plus protein, so it's just not RNA and it's just not proteins. And during the building of the ribosome, you'll also have other processing, secondary processing, that will methylate certain rRNA residues, and this prevents the rRNA from being destroyed, which would be a very bad thing. You don't want your ribosomes prematurely destroyed by your own machinery. So that was prokaryotic. Let's look at eukaryotic rRNAs now. And it's similar to how the prokaryotic rRNAs are made, in that the genes are transcribed in the nucleolus, and usually it's several hundred repeating copies of the rRNA in a row, and you'll make a giant primary rRNA transcript. 7,500 nucleotides, that's called the 45S rRNA. And like the prokaryotic version, this contains different parts of the rRNA. You have your 18S rRNA, your 5.8S rRNA, and the 28S rRNA. And between these RNAs, you have spacer sequences that won't be used in the final process. So our first step into making this 45S RNA functional is that we have to methylate it. And we have methyl groups to 106 different sites in humans. 80% of those methylations is at the second prime oxygen to make a methylribose. The rest are methylating bases. And so the question is, how do we target these 108 sites? And the answer is, our pre-rRNA will interact with small nucleolar RNAs called SNO RNAs. And mammals have roughly 200 of these RNAs, and they're 70 to 100 nucleotides long. And 10 to 21 of these nucleotides are complementary to the mature rRNA that needs to be methylated. So what happens is that this SNO RNA will bind to the site it needs to bind to, that is complementary to, And this is directing the methylation machinery to combine to this SON, SNO RNA, this rRNA, and methylate where they're located at. If we didn't have these SNO RNAs, you could think that we would need a different methyltransferase to recognize each methylation site in 45S rRNA. So we would need at least 106 different proteins just to add methyl groups, which would be highly inefficient. And along with methylation, we can also have modification of uridine. So we have some RNAs and proteins catalyzing uridine to pseudo-uridine here, and you can see the difference where we add a nitrogen at this position. Now, some eukaryotic rRNAs do contain introns as well, and the pre-RNA is one of those, but it is self-splicing. That means it acts as its own enzyme. And this is one of the pieces of evidence for the RNA World Hypothesis. The RNA World Hypothesis is that all life started with RNA, and that's because it's been shown to be an enzyme, and it's also genetic information, and it can be structural. So that's why people think RNA started all life. But let's take a look at the self-splicing event for our RNA. And it's a three-step reaction, and you need a free guanosine or a guanine nucleotide for this reaction to take place. So what happens is that the 3' hydroxyl guanosine will attack the intron 5'N as shown here. And what happens is that the 3-OH hydroxyl is now free of the exon. And this then will go and attack the other intron. So the 3'OH will go and attack the 5' of the other exon, and you'll form a new bond where you have your exon and your intron has been totally displaced. So we have made our exon, but we still need to take care of our intron. So what happens next is that the 3' OH group of the intron will attack an A residue, roughly 15 residues away from the end of our intron here. And so our 5' terminus is actually displaced, and so we make a cyclized intron with a fragment left over. Interestingly enough, this does not take energy, so this no ATP, no GTP needed, and this type of self-splicing intron is known as a group 1 intron. There's also another group of introns called group 2, where you will use an internal A residue to form a lariat. And if you remember, when we talked about intron splicing, that also forms a lariat. So what this suggests is that the spliceosome mechanism that's around for mRNA probably evolved from a primordial self-splicing rRNAs, and proteins are just there to fine-tune the ribosomal structure and function. And with that, that's the end of this video. I will see you in the next one.